Okay, so welcome to the second lecture for statistical physics of ecosystems. Yesterday, we sort of had a table setting motivation kind of thing where we talked about the kinds of things that we're trying to explain maybe in ecology, how it sort of got started, and what kinds of broad models people kind of use. And we kind of ended up at this idea of trying to focus on only one patch spatially that's well mixed with some number of species and some number of resources, no noise, no migration. And then we even try to coarse grain out the resources and focus only on an effective model of competition between species or interactions between species, right? And that's what we call the generalized Lotka-Volterra model. So in the next 4 classes, we'll sort of look at the properties of what happens to the statistical properties of ecosystems of this kind of model. And then sort of towards the end, in my last lecture, I'll sort of talk about going beyond this model. So the goal for today is mostly to talk about the GLV model in the what I call low dimensional limit, in the limit where there are 2 species. And as we sort of go on, once we sort of build an intuition for 2 species, we'll go on to asking what happens sort of immediately to a large number of species. Okay, so what we're going to start with today is to talk about first, what is it that we observe experimentally when people have tried to compete to species. And we talked about this a little bit yesterday when we were talking about the experiments of Gauss in the 30s. And really, there are 3 broad qualitative outcomes that people observe largely. One is that if you put 2 species and you get them to compete, you see exclusion. That is, one species outcompetes or excludes the other. Sometimes in some pairs of species, you instead see coexistence where both species can survive for a long time. And then in some other cases, this is not a case we talked about yesterday, but we will talk about today, you see effectively bistability, which is depending on the initial conditions. If you start off with a lot more of species A than B, then A will win. If you start off with a lot more B than A, then B will win. And these are the 3 broad qualitative outcomes that are observed experimentally. And then we'll go on to ask, what are the broad qualitative outcomes that you get from a 2-species Lotka-Volterra model, which is what we wrote down at the end of the class yesterday. And what we'll find is that this sort of model of competition with 2 species reproduces exactly these 3 qualitative outcomes. These are the entirety of the 3 outcomes qualitatively that are allowed by this kind of model. And so in that sense, it's sort of conceptually interesting. This is kind of what you see in experiments, and you need like a really simple model with effectively 2 sort of parameters, the interactions between which are how species 1 inhibits 2 and 2 inhibits 1, that really tell you everything you need to know about the behavior of these 2 species in the long term. And then maybe towards the end of the class, we'll start to generalize some of these ideas that we learned from the 2-species model and see how we might start to think of them more carefully, where S, or the number of species, is going to be larger than 2. And in particular, as we're going through the 2-species model, we'll focus on 3 things, 3 properties of these ecosystems. In addition to the qualitative outcomes that we get, we're going to worry about feasibility, which is the idea of asking, if you're looking at a particular fixed point, is it feasible or not? This is kind of an idea that maybe you're not very used to when you're thinking about other physical systems. But of course, the abundances of species cannot be sort of negative. So if we end up with fixed points of these kinds of dynamics where the abundances of one or both species are negative, that's not going to be a physically relevant solution. Right? So we're only going to care about feasible solutions where some species can be extinct, others can be non-extinct, but at positive abundances. We're also going to talk about stability. This is the first time we're going to start to think about what it means for a fixed point of these ecosystems to be stable. Some of these things you might have looked at in the preparatory part of the school, so we'll kind of jog those memories up. Again, we're going to look today at the low-dimensional versions of all of these. And pretty soon, tomorrow onwards, we're going to start looking at high-dimensional versions of these. So if you have any confusions about what we do today, please come and ask me, or please sort of brush these up, because it's going to get yeah, if you have trouble following this today, you're going to have a much harder time tomorrow. Yeah. And then finally, we'll talk about structure, which is the fixed point itself. If both species survive, which species is more abundant and which species is less abundant. For two species, this will be very easy to see. But again, as we sort of go on to asking what happens with infinitely many species, is going to become not as straightforward as it is right now. Yeah? So this is kind of getting us into the feeling of doing these kinds of calculations in a simple version of this model where we can really solve for everything. Yeah. Okay, so let's first start with what the experimentally observed outcomes are when you try to compete two kinds of species. And I spoke about this a little bit yesterday. But let me just quickly describe again what are the kinds of experiments that one usually does in this kind of realm if you wanted to compete two species that you picked out from somewhere. And so what you usually do is you kind of start off doing something that's very commonly done, which is a serial dilution experiment. Okay. And this is a very, very common experiment that's done, especially in microbial ecology. It's kind of nice to spend 5 minutes sort of thinking about how you would go about doing this. So the idea of the experiment is kind of simple. You have a test tube or a flask or whatever your favorite container in which you want to grow a bunch of microbes. This is my cartoon of a test tube. You put in some sort of medium. This medium will usually be water mixed in with a bunch of nutrients, some sort of carbon source, some sort of nitrogen source for species to grow in. And this will be some amount of some concentration of this medium that you will provide that allows for growth. And then you also should have two species that you actually want to compete, right? So presumably you've gone out in the world somewhere, isolated two species. I won't talk about how you go about doing that, but say you've done that. You have species 1 and you have species 2. What you do is you put a little bit of both these species into your test tube. So they go in, and what would happen if you just kind of leave this be? I mean, the next part of the experiment is simple. You go home, right? And so what do you think will happen once you go home to this test tube? Sorry? No one knows. All the microbes will be kind of sitting there doing nothing. Right, yeah. So the microbes will grow because there's a bunch of food in there, and as they sort of start to grow, they'll eat up these sort of nutrients, and maybe both these microbes are probably eating the same set of nutrients that are present in the medium, so they'll start to compete with each other, right? And do you think this will go on forever? Do you think they'll just keep multiplying? Yeah, at some point of time, this process will stop, right? At some point of time, they'll run out of nutrients. So you'll have a whole bunch more microbes of each type than you maybe started with. But at some point, this thing will stop, right? And so you have to come back from home to the lab. And what do you do now if you want to continue? If you like, do you think we're done? If we just went and asked what fraction of species 1 there is and what fraction of species 2 there is, we would know exactly what happens at the end of this competition. Why not? There, no, you put in a bunch of species, you grew them out, they grew, so maybe 1 grew more, 2 grew less. And this process stopped. So maybe you have a way after that of taking out some of these microbes and measuring essentially by some fancy version of counting what fraction of species 1, you'll kind of, what you typically do is you take out a little bit of the sample, you plate it on something, and you'll start to see some spots, and you can kind of tell if you're a trained microbiologist what spots belong to species 1, what spots belong to species 2. So this might be 1, 1, 1, this might be 2, 2, 2. And yeah, you can say that there are 3 out of 1, 2, 3, 4, 5, 6, 7, 8. 3 out of 8 roughly is the fraction of the population of species 1. 5 out of 8 is species 2. What might be the issue with this sort of number? Or maybe there's no issue. 3 out of 8, species 1. these kinds of numbers of the population or these numbers? Yeah, these numbers, you don't have to worry about being very small. I'm just kind of lazy to draw out. People are not counting 8. People are usually counting hundreds. And you can usually back calculate to within 5 or 10% error. And 5 or 10% error is actually pretty good because the population sizes here are, like we discussed yesterday, in the billions per ml, right? So 5% error on that is really very small. So the numbers are not really a problem. This will usually not be 3 out of 8. It will be more like, I don't know, 34 out of 169, right? So it will be something of this kind. So you don't have to worry about the accuracy of these numbers. Yeah, maybe you should repeat it, but that's really for reproducibility. I think my question is really, would you then believe that, like, when these two species compete, they coexist? That's what this outcome is telling you, right? Yeah, so fine. So you might, but in the end, if I care about, like, these outcomes that I care about are all qualitative long-term outcomes, right? They're all outcomes that I care about after a long time. So yeah, you're right that I don't see the transient. And maybe if I saw the transient, I would then draw a picture of how I go from 50-50 to 34-169, right? I could do that. Let's say I could do that. Would I then could I then say confidently that these two species coexist? Mm-hmm. Mm. Yeah. Exactly. So you're saying that, it might be that the competition is actually in the midst of playing out, and you don't know what's going to happen because the resources ran out before it could play out, right? That's true. So you kind of hit pause in the middle of the movie, right? So how would you? Yeah, that might be the case. So in other words, what you're saying is that this coexistence might not be stable, right? When you want coexistence to be stable, you want it to be the case that, of course, every time you do the experiment, you see the same number. But of course, every time you start the same movie and you stop it after 47 seconds, it will look the same, right? But that's not to say you finish the entire movie, right? So you need to do something to kind of let this movie play out, right? And it's only the case that if you let it play out sufficiently that over a long time you saw the same number, not for many replicates, but in the same culture, if you saw the same number stabilize, that's when you would know that you've taken the competition to its logical conclusion, right? So just because, because how do you know how much medium you put in, right? You could have put in a little bit, you could have put in a lot. Okay, so what do you do to like press play on this movie again? The resources have run out. So would you put the resources back in here? Because you want everything to be clean or something, is it? Yeah, so I mean, you do use a new test tube. And then what do? I mean, this has fresh medium, let's say. You put in fresh medium with a fresh batch of nutrients. And then what do? Do I transfer this entire thing over to the test tube? What will happen if I transfer this entire thing over to this test tube over here? Sorry? The volume increases. Okay, let's say that I could somehow filter out the cells from the medium and only transfer the cells. Would that be a good idea? Okay, why a little bit of it? Okay, so that's one point, that if, of course, if I took a small fraction of this whole pool, like the numbers are so large, I'll probably still sort of preserve the fraction. But why do I want the numbers to become small here, the absolute numbers? Exactly, right? I mean, if you put in the same number of cells, they'll probably need a lot more nutrient, right? So it's like you hit pause on your movie before, and you only play it for 1 second before it stops again, right? So you kind of want a situation again where the number of cells initially is much smaller compared to the amount of nutrient that you have, so that this movie can run a little bit longer, right? You kind of don't want to come back in 5 seconds again and do this whole thing again, right? So what you do is you take a little bit of what was in the test tube, large enough that, again, remember, there are billions of cells in here. So even if you take 1%, that's 1% of a billion cells, right? So the fractions are probably going to still be preserved. There's going to be some Poisson error, but that Poisson error is, you can imagine how small that will be, right? You transfer that over in here. And what fraction you take over here is going to be determined by some factor D, which is the dilution factor. That's a factor by which you basically dilute this population over to the next medium. And then, you put a little bit of cells again, and they'll grow out, and then you can count them, and then you do this again and again and again, right? So what you're doing is effectively you're doing two things. You're doing dilutions of a population, and you're doing it in serial, right? One after the next, after the next, after the next. And by basically counting, usually you choose a nice enough time as an experimentalist that you're waiting 24 hours in between this. So you're coming in at whatever, 9 a.m. every day, doing this experiment, spending 5 minutes preparing this fresh medium, diluting this out, and then going home again, right? And you can do that over many, many days. Every day you take out a little bit of the sample that you left from the previous day, you count this out, and you can track fractions over time, right? And that's how you can get essentially quantitative estimates of the fraction of species 1 versus species 2 over time in this two-species system. You can also do it now over many replicates, as you were saying, and kind of ask how reproducible the outcomes that you see are, right? And what do you see? Let me just draw this before I take the question. So what you make is a plot very similar to what we were making yesterday. You plot time in the number of days because you're coming in every 24 hours and doing this measurement. So it's a very convenient unit of time from a human perspective. And on the y-axis, you plot simply this fraction of species 1 or species 2, whichever you want. By the way, I'll keep saying SPP for species. I hope that's okay. Okay, and this fraction, of course, can only take a number between 0 and 1, right? That's what fractions are. And if you do this experiment, this one go, the serial dilution experiment, basically you'll get 1 point at t equals 0. That's the initial fraction that you kind of mix these two species in. And then basically every day you'll get a new point. And sometimes if you do this experiment, this is what you might see. I'll just connect these points to make it visually easy to see. What would your interpretation be of what this experiment eventually leads to? Okay, let's take a vote. So we'll have our 1, 2, and 3. Those are our fingers. We'll say exclusion is 1, coexistence is 2, bistability is 3. Okay? I'll count to 3 just to remind you what we're doing. And everyone will, I hope everyone will, vote 1, 2, or 3 based on what they think is the outcome of this experiment if you did it. All right? Ready? 3, 2, 1. Okay, yeah, basically I would say the majority of everyone thinks it's exclusion. And can someone say why? Which species excludes which species? 1 excludes 2, right? So this is an example of exclusion where 1 wins. And of course, there was a question about how reproducible this outcome is. So you can maybe start off with a different initial fraction, and you might see the same thing. Might start off with a rather different initial fraction. You might start off with the same fraction that you started off with, and just do that experiment again. This is a very easy experiment to do in the large scheme of things. And what you might see is that again and again and again, of course, it takes a different amount of time based on the initial fraction for you to get to this eventual outcome. But eventually, you always see species 1 being 100% of the population, which means visually, at some point as an experimentalist, you'll go plate this sample, and all you'll see will be these like hollow colonies, right? Of course, you can increase the resolution further and further and try to kind of make sure that really there's no species 2 out there at some very low resolution. Yeah, okay, sorry, there were a bunch of questions that I did not take, so I'm ready to take them now. Yeah. Good. Yeah. So it's the fraction of the numbers of cells that you are moving to the next test tube. That's a very good question. So typically, again, you want your experiments to be simple. And so for the large majority of experiments, a lot of these parameters are constant for a given experiment. The dilution factor will be constant, or it'll be a number chosen by the experimentalist. The time between dilutions will be a constant, usually some multiple of 24 hours for obvious reasons. The amount of nutrient in the medium that's provided every time is also a constant chosen by an experimentalist. What else would be a constant? I mean, you'll try, of course, you try to make the rest of the conditions of the laboratory as constant as possible, temperature, where you do the experiment, et cetera, et cetera. Yeah. Very good question. How do we determine the time scale of this measurement? I would say a little bit, this depends. Okay, I mean, experimentalists will have a good sense of this, and the way that they get a good sense of this is because you typically try, when you isolate these species, you try and grow them out in different media on their own in the absence of other species. So you, and then you kind of track their population sizes on their own over time. You get a sense of what the growth rates of these populations are. And so what you want typically for this competition to play out is you want a certain number of doublings to happen, right? In every sort of run of this experiment, in every dilution step. And so you kind of work backwards given the growth rate and the initial concentration of the nutrients, how much nutrient you should add, how much, and yeah, basically whether you should wait 24 or 48 hours. So like I said, it should be, usually it'll be some multiple of 24 hours, right? For reasons of experimental convenience, but whether it's 24 or 48 might be determined by whether species are fast growing or slow growing. If they're slower growing, they might not be able to exhaust all the nutrients in 24 hours. And so you wait longer. Do we need to, that's an excellent question. Yeah, do we need to take care of minimum population size? Again, we discussed this alley effect yesterday, which is about when you're growing a single species, you might need a certain minimum population size. And so indeed, like I said, before you compete two species, you'll try to figure out what they do on their own. And so at that point, you'd want to grow the species on their own at different starting densities and figure out basically whether or not they have this alley effect or not. And if they do, then you're exactly right. You want to make sure you choose a dilution factor such that this bottleneck population size is larger than the one that's above this threshold of minimum population size. So that's exactly correct. And it might be, of course, once you choose this initial dilution factor this way, it might be that, of course, because of competition, the population size of the other species might be driven down so low that once you dilute it over, it might go below the threshold. But that's actually an outcome of the competition, right? Yeah. How are we deciding the nutrients? Yeah. Yeah, you're absolutely right. So, I mean, of course, one way you choose them is if you, like, one thing you do, of course, is you make sure you're not choosing like a boring nutrient, right? Which is a nutrient in which one or both of the species do not grow on their own, right? So, of course, that automatically like kills out a whole bunch of nutrients. So, like I said, you'll do a lot of characterization usually before you compete species, what they like or don't like on their own. And you'll try to grow species together in the same medium. And you'll try to choose a medium, of course, in which both species grow. And it's not totally obvious that one species has a clear advantage over the other. Though you will be surprised that there are many cases in which actually it seems that when you grow species on their own, one species is like twice as fast as the other, but they can still coexist when they sort of grow, when you sort of grow them together. Or there might also be cases where the slower growing species actually outcompetes the faster growing species. Exactly. So initially you try out to grow these species in what is called monoculture, meaning that you culture them on their own. And if they grow well in nutrients in monoculture, then you choose some subset of those nutrients in which both species can grow. Exactly, because yeah, so the question is, it's probably hard to take out the microbes, so maybe you just take a little bit of literally the filtrate. The rest of the filtrate should probably just be water, because the nutrients you expect to be exhausted, and then you kind of move it over, and that should have a minimal effect. That's indeed true, though I'll say two sort of addendums to this. It's actually not that hard, as far as I understand, to extract these. You just need a fancy filter, like a channi, basically, with just a really fine one that keeps the cells on the top, lets all the stuff flow down. The microbes typically, yeah, bacteria will typically be about 1 to 2 microns. Yeah, so you can imagine between 1 to 10 microns or so. You can have sieves for those. And then the second thing that I will say, which maybe I shouldn't say it because that will confuse you, but I'll talk about it in my last lecture. This will typically have about 1 billion cells per ml, and the culture volume of this flask will typically be about 10 ml. Yeah,
but this number is 169, that's because of the way you count.
That's just, yeah, basically what you're doing, yeah, great question, is before you count, you will successively dilute this many times fold, just so that you actually are not counting billions of cells. So you know the factor of like some power of 10 by which you diluted this by. And then if you see 169, then that's 169 times 10 to the whatever factor you dilute. Okay, great. These are all great questions. Okay, excellent. Okay, so that's one qualitative outcome that you might see if you compete two species. It's not the only qualitative outcome that you might see. So if you do it again for another pair of species, 3 and 4, let's say, what you might instead see is something of this type. I'll try not to be not discrete in what I draw, but it's in the interest of time. This is what you might see. Okay, and what kind of outcome out of the 3 is this one? Let's take a vote. Let's not say it out loud first. Okay, ready? 3, 2, 1. Yeah, it's coexistence, right? And you can kind of see that it's a very nice kind of coexistence in that it's stable, right? Which is to say, you might arrive at maybe not a very stable coexistence where whatever fraction of species 1 and 2 you started with is roughly the fraction at which you stayed, right? That's, I guess, neither stable nor unstable. That's kind of a marginal. But in this case, whatever initial fraction of species 1 or 2 that you start off with, you kind of over time eventually go to a well-defined stable fraction at which species 1 and 2 will coexist. Yeah, I hope it's clear what the axes here mean. It's the same thing. I'm just not writing it down because I'm lazy. But I hope it's clear kind of what's going on. Yeah. So each trajectory here is one experiment done over many, many days, roughly about 2 weeks. And every independent trajectory here is a different replicate where initially you mix species 1 and 2 at a different fraction. You can choose when you initially inoculate that whether you put more of species 1 or 2. Yeah. Everyone's clear? And I'm also not drawing error bars for these, but you can imagine that the error bars come from just Poisson counting error. Yeah? Okay, so here you might see a situation where species 1 and 2 stably coexist, and of course they coexist at some fraction, maybe that you might call F star, and we might care about what that fraction is and why. We'll talk about that later today. And the third and final outcome, which is the other kind of outcome that we see, is called bistability. So what do you think happens in bistability that's qualitatively different from this and that? Exactly. So visually, what should I see here? Basically, some kind of forking out, right? I start off here. I'll see this. If I start off below some threshold, I see something like this. And basically, there's some other fraction, F dagger, let me call it, which maybe if I were to start off at perfectly, even though I will not be able to, maybe I would see some sort of coexistence, but you won't, right? So basically, there's some threshold frequency or some threshold fraction of species 1. You start above that, species 1 wins. You start below that, species 2 wins. Yeah? So this is the region where 1 wins. This is the region where 2 wins. And so this is a situation in which there are 2 stable outcomes, right? Either 1 wins or 2 wins. That's why this is called a bistability. This is also something that's commonly observed. And there's really no 4th outcome that you usually see, but can you imagine what other outcomes might be like, just so that it's clear that maybe it's not trivial that, of course, you should get only these 3 things and nothing else. Can someone think about maybe a 4th outcome that could be possible, like a 4th way of making these plots, but that actually doesn't happen? Sorry? Both populations oscillate. That could definitely be the case. Typically, if you grow microbes in pairs, that's not what you see. But of course, predator prey and stuff like that, we've kind of all seen. Both die. Yeah, that definitely could be the case. But the question is, do both die because you did something silly, like you didn't provide the right nutrients, or do both die because, well, they both kind of toxified themselves to death? It might, you might imagine it happens. Okay, anything else? Good, right. So right now we are imagining that these species stay the same during these entire dynamics. Microbes might mutate, that might happen. And again, that might do something weird. We're not going to worry about that. Anything else? What about if, like, can this curve look anything different? I don't know, when I first saw this, I was like, oh, why does there need to be just a bistability? Why can't there be a tristability? Or why can't there be a bistability where in one stable state, one species wins, in another stable state, both species coexist? It's not impossible, right? It didn't have to be this way, right? So again, I'm just trying to tell you that there are other possibilities that could have happened that, at least in terms of curves, are not disallowed. That's usually not what you observe. There are actually one outcome that people have seen where you do observe this bistability where one species wins or both species coexist, but we're not going to worry about that peculiar outcome because that's something peculiar about the biology. Okay, so these are really the kind of outcomes that you see. This is kind of typically how you would do or set up competition between 2 species. And then the rest of the lecture, I kind of want to spend time talking about how and whether the 2 species Lotka-Volterra model can capture all of these 3 outcomes. And so my answer is going to be the following, that it is going to be able to capture it. And in particular, we'll spend the rest of this class really focusing on this crude kind of phase diagram of the Lotka-Volterra model, or where these outcomes lie. Just to remind you, the 2 species Lotka-Volterra model concerns writing down the dynamics of 2 species with abundances N1 and N2 in a single patch with no noise that interact with each other. And yesterday, we came up with this idea that we're going to write down a simplified version of these dynamics of the following form. So remember, in the general version of these dynamics, there were R's sitting here, there were K's sitting here, and we were working in the limit where all the R's are 1 and all the K's are 1. And so now really, how many parameters do we have in this model? Where again, remember, we'll use our fingers, yeah? How many parameters do we have in this model? Ready, 3, 2, 1. Okay, yeah, so most everyone thinks it's 2, right? And so it is indeed 2, in which 2? There's really just the interactions, right? A12 and A21, right? In principle, the full model would have had, 6 parameters, but we kind of threw those out. And so really, this kind of diagram that we're going to draw here is really going to be concerning the values of these 2 parameters. The way I'm going to draw it, you'll see quickly in some time why. I'm going to think about the log of both of these. So this is log A21 and log A12. And to remind you, A21 means how species 1 is inhibiting 2. A12 is how species 2 inhibits 1. Right? And we're also working in a competitive version of this model. So what are the signs that we are thinking about for these A's? Is A positive or negative? Ready? 3, 2, 1. Okay, people seem to be confused. Half of you seem to be saying it's positive. Half of you seem to be saying it's negative. Maybe spend 10 seconds looking at your neighbor and trying to convince them which is what. Okay, all right. So let's gear up to take a vote again. I hear the talking dying down. So does competition imply both these A's are positive or negative? Ready? 3, 2, 1. Some people decided not to vote anymore, but for those of you who did continue to vote, yes, indeed, both of these are positive. Right? That's because with the sign convention, the way that we write these models, this is already negative. And so the more and more of each of these species inhibits the growth of the other one. Right? Okay, and so really what we'll see in the rest of this class is that based on the values of A21 and A12, we're going to have these 4 or 3, based on how you count it, outcomes that are possible. Just to orient you, what point here does the origin mean? 1, 1, right? So this is just logs of both of these interaction parameters. You'll again see why 1, 1 is the right comparison. But if both of the interaction strengths are larger than 1, then you get bistability. Both of the interaction strengths are smaller than 1, you get coexistence. If the inhibition of 1 on 2 is larger than 1 and the inhibition on 2 on 1 is smaller than 1, then there's exclusion and 1 wins. Makes sense. And if it's the other way around, then there's also exclusion and 2 wins. Okay, so it's really about the interaction strengths and what they are compared to the scale 1. Okay, and where does the scale of 1 come from? Sorry, can you be a little bit louder? Kappa. What's kappa? Right. So you're thinking that it comes from this carrying capacity. Okay. Why would that be? So what units would carrying capacity have? Yeah. And yeah. Okay. So yeah. What's your logic for why it should be the carrying capacity? Okay. And yeah, the rate could be scaled. But then why would the right comparison be this one? Okay, so we'll see. I'll talk more about this in a bit. But the right comparison turns out not to be the carrying capacity, but instead the self-inhibition of this species. So remember here, this number over here is also 1, right? And what is this? This is in some sense A11, right? This is how species 1 is affecting itself. And remember, like every species limits itself, because as you eat up resources, you'll deplete those resources and you'll affect your own growth, right? And this choice to make it 1 was something that we did in our sort of normalization, the way that we did it, right? So we chose this number to be 1, and sort of everything else is measured sort of relative to that. And same here. We kind of chosen a model in which all so-called self inhibitions, how species 1 affects itself, 2 affects itself, are all 1. Okay, so this turns out to be the right comparison. And if you're again confused by why that is, you'll see in a second as we try to play with this model. Okay, so let's see how to sort of see this or sort of confirm this. And so let's start off with doing one case, and then we'll talk about all these various things, and then we'll do a few of the other cases. Yeah, and then whatever case we can't do in today's class, I'll leave for homework. It'll be pretty straightforward for you to do. Okay, so I find the case of coexistence very interesting. So let's discuss the case of coexistence. And so what does coexistence require? A12 to be, is it negative? This origin is 1, 1, right? So it doesn't require it to be negative. We need A12 to be less than 1 and A21 to be less than 1. And that's what we're going to assume. Okay, and so now we need to start talking about what do we expect as the sort of long-term behavior of this two-species model if both the A's are less than 1. Okay, and so I guess what we're going to do over these classes is whenever we're going to write down a nonlinear dynamics model of this kind, we're going to talk about a few different things. The first thing that we're always going to talk about are the fixed points of these dynamics. Okay, and in that, the first thing that I will always ask you, and I will ask you right now, is how many fixed points do you think this model has? Okay, I'll give you 10 or 20 seconds to think about it, and again, we'll vote because this is a number. The question is, how many fixed points? I won't give you options as 1, 2, 3, 4. You all know what those numbers are. So first, think about it on your own for maybe 10 or 20 seconds, and we'll take a vote. And based on how the vote goes, we'll see if we all want to talk to each other. Yeah, so the question is if this is a competitive model, so both A's are positive and less than 1 because that's the situation we've imagined, right? So both the A's are somewhere between 0 and 1. Yeah, then this diagram does not apply, right? So this diagram only applies for the model of competition, but that's fine because competitive outcomes are all we're trying to explain. All right, so let's take a vote. How many fixed points do you think there are in general? Ready? 3, 2, 1. Be a little bit prouder of your hands. If you do it like this right over the table, then I really can't see. Sorry. Okay, so there seems to be some confusion between 3 and 4. Maybe turn again to your neighbor and talk about the people, especially who can count 4. Point out all the 4 to your neighbor. All right, okay. Let's try to convene back and see how many fixed points everyone can count. Maybe it went over 4. Maybe someone saw 4, someone saw 3, but then they each saw some that the others didn't. All right, so let's take a vote again. Ready? 3, 2, 1. I would say 75% of the class is just not interested in voting. And I'm really not, I'm not saying this in hilarity. Okay, let's try it again. Ready? 3, 2, 1. Okay, all right. So there still seems to be a mixture between 3 and 4. Maybe we can have people try to point out some of them. So I've made like a face portrait here for N1 and N2 so that we can mark out our different fixed points. Maybe can someone start us off? What's one really simple fixed point that we can mark out immediately? The origin, right? 0, 0. 0, 0 satisfy these dynamics kind of immediately does, right? Okay, so that's one. So there's at least one. What others? Sorry? 1, 0. Okay, let's see 1, 0 satisfies it. So 0 here, so that's all good. That makes n2 also 0, and then n1 is 1, right? So 1, 0 satisfies it. Okay, good. Anything else? Immediately by symmetry, 0, 1. Let me just mark it out a little bit lower. Okay, and is that it? Are any of these points coexistence? No, right? I mean, kind of you already know that maybe you should be seeing coexistence here. So maybe at least there's one more, right? And is there one more or two more or three more? Like how many points can there be coexistence at now? Why is it one? Intersection of some two lines. Which two lines? Okay, so you're saying that the things in the parentheses are equations of lines in n1, n2 plane, and somehow the intersection of these lines should give you that. Why? Because when they both intersect, both these parentheses will be 0, right? And because it's a linear equation, two equations, two unknowns, just one solution, right? So indeed, there's going to be one more fixed point somewhere here, which I won't mark out because I'll talk about the lines in a second, which will be actually the coexistence fixed point. And you can actually compute. Yeah, did anyone compute what the fixed point abundances should be? A21 minus 1 or something else? Usually it will turn out to be something like this. So there are 4 fixed points. There's 0, 0, 0, 1, 1, 0. And the final one, if I did it correctly, you should point out if I'm wrong, is 1 minus A12, 1 minus A over 1 minus A12, A21, 1 minus A21 over 1 minus A12. If you didn't get this, then either I did something wrong or just check your algebra. It's a small enough point that I won't bother sort of talking about this too much. But that's the population of N1. Often at fixed points, we'll make these stars. So that's N1 star, and that's N2 star. Okay. All right. So there we have 4 fixed points. The fourth one I haven't marked out, but I will soon. I mean, it'll be somewhere here. And I've drawn these sort of big open circles here for each of them, right? What am I anticipating? What should we ask as soon as we know what fixed points we are looking at? Stability, right? Not all of them might be stable. Okay, and so let's start looking at stability, and maybe let's start looking at stability one by one to make things easy. So what about this 0, 0? Do you think it's stable or unstable? And how will you tell? Let's take a vote. Stable, thumbs up. Unstable, thumbs down. Okay, ready? 3, 2, 1. Okay, I would say majority people who are responding at least think it's stable. Why do you think it's stable? There's nothing to begin with. Is that how we probe stability? What's a typical way to probe stability? Yeah, you perturb the system a little bit and you see if it comes back, right? If you find any one direction in which it does not come back, then it's unstable, right? So if we found even one direction in which this point, which we perturbed this thing and we went away, this would immediately be unstable, right? Or at least not stable. Okay, so can you think of one direction in which, okay, so do you think all directions are stable? What about if we perturb a little bit along the x-axis? We had a little bit of species 1. Sorry? Where is the alley condition in these equations? Nowhere but. Are we analyzing these equations or are we analyzing? Yeah, now we're not thinking about the world for a second, right? They're like microbes wearing pink hats and so on and so forth. We're not worried about any of this, right? We're only thinking about the behavior of these equations, right? So is there any alley effect in these equations? No, right? So let's go back. What do you think, where will the flow be if you went a little bit to the right? To the right or to the left? To the right, right? And why is that? There's no species 2. This is just logistic growth for species 1, right? Logistic growth has one and only one stable point, which is just the carrying capacity, right? So along these directions, you're stable. But immediately you can kind of see that this point automatically becomes unstable, right? Also, by the way, for this direction, right? You add a little bit of species 2 and you run away. Okay? That point is unstable. I'll add a U next to it and keep the circle kind of open. If it were stable, I would kind of fill it up. Okay, what about this point over here? This looks pretty stable, right? So let's take a vote again. Was this 1, 0? Is 1, 0 stable or unstable? Ready? 3, 2, 1. All right, everyone thinks it's stable. Anyone thinks it's unstable? Yeah? For N2, yeah, go on. Very good. So the correct question is not just to ask in the direction in which you know you come back, but to ask in every possible direction, right? So the easiest possible thing to imagine is you add a little bit of N2, you go just a little bit above this, and then you ask whether or not, yeah, what do you ask? Whether N2 grows or not, right? If N2 dies, then you flow back. If N2 grows, you go up and you're unstable, right? And so this, in the language of biology and ecology, has a very famous name that's often something that you'll hear, is something called invasion analysis. Easy to imagine why you might be doing this. You're adding a little bit of species 2. You add species 2 and you ask for N2 dot. Okay, so N2 dot, we know what our equations looks like, is something like that. And you ask to invade with species 2 right when you're already at this point, right? You're at this fixed point. You're probing the stability of this fixed point. So what can you say about N2 dot? Or rather, like, what do you want to know about N2 dot? You want to know the sign of N2 dot, right? You want to ask whether it's positive or negative or 0, right? So N2 itself will, I guess, always be a positive number, right? You'll always add a little bit of species 2. So really, the only thing that you need to care about for the sign is what's inside this parentheses, right? So what can you say about this term in the parentheses? When N2 is small, remember N2 is very, very small, much, much smaller than its carrying capacity. So N2 initially is much, much less than 1. So what can you say about this term in the parentheses? Can you simplify it? What is 1 minus N2? 1, right? And what is N1? Is N1 small? You're probing the stability of this point, right? So this is 1, 0. Is 1 much, much smaller than 1? No, right? So you replace N1 with 1. Okay, I'll erase this bit out. We ignored N2 because N2 is very small compared to 1, right? And so what do you end up with? All you care about is 1 minus A12, right? And what's the sign of this guy? A12 is less than 1, so this is greater than 0. And so this species will grow, right? Species 2 grows, or in the jargon of ecology, it invades. And that means you have at least one unstable direction that you found. That's unstable. What about 0, 1? Same logic, right? You add a little bit of round species 1, right? You do the same thing. N2 dot becomes N1 dot, becomes N1. This is 1 minus N1. This is 1 minus A21 N2. All the 1s become 2s. What do you have? You have again that N1 is much, much less than 1 because you're invading with a little bit of it. So you neglect it. N2 at this fixed point is 1. You substitute as 1, and again you end up with 1 minus A21, which again you know is going to be positive. So species 1 will grow and invade. And so again you found one more unstable direction. So this is also unstable. So right now it's looking pretty bad for us, right? We found 4 fixed points and we kind of killed 3 of them because they were unstable. So we better hope that the dynamics lead us somewhere, and maybe this point that we found here in the center is stable. So let's just sort of talk about that. Okay, so thus far we've only kind of been plotting these points based on what we knew about the equations, right? But if we were really trying to look at this point over here, we would also typically care about these things called null clines. Right? Are people familiar with what null clines are? Okay, people are not familiar. That's fine. We can talk about null clines now. So really what we, like, a fixed point requires what, by the way, in these dynamics? N1 dot should be 0 and N2 dot should be 0. If only one of them is 0 but the other not, is that a fixed point? No. Right? This is a very, very common mistake that I see students do all the time. It becomes even more common when there are not 2 species but infinitely many. But if just one species is not growing, but the others are growing or dying, that's not a fixed point. Fixed point is a property of the entire dynamics. Okay? So the dynamics, for them to get to a fixed point, requires both of these quantities should be 0. But you can often ask first about each of the individual degrees of freedom being not growing or dying. You can ask for N1 dot equals 0 or N2 dot equals 0. Right? It's only when both of these conditions are satisfied simultaneously will you end up with fixed points. Right? So what does N1 dot equals 0 give us? It gives us curves. Does it give us one curve? How many null clines are there? How many curves are there that satisfy N1 dot equals 0? Ready? 3, 2, 1. So some people think 2, some people think 3, some people think 4. Maybe talk to your neighbor and discuss. Okay, all right, let's take a vote again. So N1 dot equals 0. How many curves is that? Ready? 3, 2, 1. Great. Okay, excellent. There are 2 curves, right? In which are they? Either N1 equals 0, which is the y-axis, right? So if I'm just going to try and draw in orange these null clines. It's either the y-axis. I hope I got that right. I always mix up the x and y axes. Okay, and then what's the other one? N1 equals 0 or what? N1 equals 1 minus A1 to N2, right? That's that term in the parentheses. So that's the way I've written it is 1 minus A1 minus A1, 2, and 2 equals 0. And that's a line. That's an equation of a line in N1, N2 plane. And just to get a line, we just need 2 points. So when N1 is 1, N2 is 0. So that's 1 point on this line. And when N2 is 0, sorry, when N2, sorry, and when N1 is 0, N2 is 1 over A12, right? And is 1 over A12 above 1 or below 1? Above, right? Because A12 is less. There's 1 point here, and there's 1 point here. That's our line. Okay? That's 1 over A12. Okay, these are our two null lines for species 1. What about for species 2? Again, there are 2, right? There's N2 equals 0, and there's 1 minus N2 minus A21 N1 equals 0. That again, for species 1, that's the x-axis. And then the other one is when N1 is 0, N2 is 1, and when N2 is 0, N1 is 1 over A21. And is that to the right or to the left? To the right, because A12 is less than 1, right? That's 1 over A21. Here's that point of intersection that we were talking about previously. Okay? And so what happens at this point? They're both satisfied, so it's a fixed point. Okay, can we see that all the other fixed points are also satisfying this thing? This is a point where an orange and a green line meet, the fixed point. This is also a point at which an orange and a green line meet. And this is also a point where an orange and a green line meet, right? This is a point at which an orange and an orange line meet, not a fixed point, right? Same here, that's where 2 green. Right, so indeed we kind of verified that there are 4 fixed points here, and with 4 sets of lines that's what you might expect. And so this is our coexistence fixed point. Great, so we found that fixed point. How do we assay the stability of this fixed point? Can we do that same thing that we did before, where you add a little bit of one or the other? Seems a bit hard, right? So is there anything else that you guys learned, maybe in the prep part of the course or in something else, about how you figure out the stability of fixed points in general? Taylor expand what at which fixed point? Taylor expand the dynamics around the coexistence fixed point. Okay, good. And check the matrix. Yeah, right. Does someone know another name for a linear stability matrix? Yeah, typically a Jacobian, right? So what you're doing is that you're going a little bit distance away from this fixed point, right? You typically, if this is n1 star comma n2 star, you go n1 star plus delta n1 comma n2 star plus delta n2, where delta n1 and delta n2 are small. It could be negative or positive. And then you look at the stability of this fixed point by essentially linearizing the dynamics. You sort of Taylor expand, and you end up caring about the properties of one special matrix, which is called the Jacobian. Yeah? Can I write over on this side of the board, or is that bad? Okay, so this is fine. Okay, so maybe I'll write it down here. So we want the Jacobian or the linear stability matrix for this model. And this is good because we'll use this Jacobian in the rest of our classes as well. So let's just try and figure out what this Jacobian should be. So does everyone know what you're actually doing? What you're really doing in some sense is Jacobian J. You're essentially taking partial derivatives of each of these equations with respect to each of the degrees of freedom. You're taking del n1 dot over del n1, del n1 dot over del n2, del n2 dot over del n1, and del n2 dot over del n2. And in particular, this is just a function, right? You kind of want this to be a matrix that's evaluated at some point. So what point do we care about evaluating this Jacobian at? At the coexistence fixed point that we care about, right? Which is this thing that I wrote over here. So I'll just call that n vector star for convenience, right? But it's n1 star comma n2 star, which is that entire thing. So let's just sort of do out this partial derivative just for us to get a sense of what it sort of looks like. So let me just do one. Let's just do del n1 dot over del n1. What should that be? You know the equations for the model, and you're all better at taking partial derivatives than I am. What should that be? This is not a trick question, right? Just have to take a partial derivative of this guy with respect to n1. Yeah, so first, I guess you first do this n1 bit, and you get 1 minus n1 minus a1 to n2, right? And what would this evaluate to if we were doing it at the coexistence fixed point? Are people confused? What's happening at the coexistence fixed point to this parenthetical term? It's 0, right? That's exactly how we got this logic for this coexistence fixed point. n1 is not 0, n2 is not 0. So the only way n1 dot and n2 dot can be 0 is if each of the terms in the parentheses is 0, right? So there's one part of the partial derivative, which is that 1 minus n1 minus a1 to n2, which when we evaluate at the coexistence fixed point should give us 0. Is there any other contribution? What's that? Minus n1 star, right? Do people agree? That's also what I got. I'll write it down. Okay, well, let's do maybe one more so that we're feeling okay. Let's do the second bit here, del n1 over del n2. What's that going to give us? Minus a12 minus a12 times n1 star. Right, and you can kind of see where this is sort of going. You can, if you sort of do the other ones as well, you'll find that basically you can write down the Jacobian by factorizing it into these two matrices. You'll end up with n1 star, n2 star, 0, 0. That's a diagonal matrix which contains our fixed point. And then this is another matrix. And what does this look like? I mean, let's just verify that it's correct. At least it seems correct for these two things that we evaluated. So the first entry seems to be minus n1 star. And the second entry seems to be minus a1 to n1 star. Right? So you can also see this, the row over here will be minus a21 n2 star and minus n2 star. Right? And I've factorized it out the Jacobian this way because it really has this nice factorizable bit, right? So it's really negative of this first matrix, which is just a diagonal matrix which contains your fixed point and star, right? You put on all the diagonals n1 star, n2 star, n3 star, and so on and so forth. You can, I mean, nothing will change if you were to do this now for S species. Right? You can also verify that as homework. And this bit here, this also looks like a special matrix, right? What matrix is this? This is the interaction matrix, right? Remember, all of the diagonals here are 1, and that's exactly what we talked about here, right? All the diagonals A11, A22, and so on and so forth are all secretly 1. This bit here is the interaction matrix. Right, so you can kind of see that the interaction matrix is a really important part of the Jacobian. And so it really determines the stability. It's really important for determining the stability. The Jacobian is not exactly the interaction matrix. It has this bit over here, which is a diagonal bit which contains the fixed points themselves. But this is in general, this is the Jacobian for the Lotka-Volterra model at any fixed point. Okay? All right, so now, okay, we found the Jacobian, that's great. We wanted to assay the stability of this fixed point. What do I do once I have a Jacobian to do that? Find the eigenvalues of this matrix, okay? And what should I expect? If the matrix is stable, what should I want the eigenvalues to do? Both of them should be negative. What if they're complex? They could be complex, right? So then should some special, the real part of both the eigenvalues should be negative, right? And again, if you're confused about why that should be, look it up or come and ask me after class, yeah? Okay, and then of course, people also know that for a 2x2 matrix, it's pretty easy to actually tell stability, not by computing eigenvalues, but just looking at properties of the Jacobian matrix. Which two properties do you look at? The trace and the determinant, right? And what's the idea? So of course, the trace is like the sum of the eigenvalues, right? And then the determinant is, yeah, like the product. Here, typically you might remember, you don't need to, you could have even used that thing that we just discussed to get that. But if you have trace and determinant, usually this bit over here is unstable. So if you have a positive trace and a positive determinant, that's really unstable. Positive determinant and negative trace is stable. And over here you get saddles, which are also basically not stable, right? So where are we in this diagram for the two species Lotka-Volterra model at the coexistence fixed point? What's our trace? Minus n1 star plus minus n2 star, right? n2 star is positive, n1 star is positive, so that trace is negative. Okay? That on its own is not enough to tell us whether we are stable or at a saddle, right? So what about the determinant? What's the determinant of the Jacobian? Again, you guys are much better at algebra than I am, but if you do it out, you will see that it should be n1 star, n2 star times 1 minus a12, a21. Okay, and again, we know a12 and a21 are both less than 1, so their product will be less than 1. So 1 minus something less than 1 is positive, n1 star is positive, n2 star is positive, so the determinant is positive. And so this is where we are, negative trace, positive determinant, and so we're stable. Yeah, there's plenty of other ways you could, if you want, just go at it and compute the eigenvalues from the matrix. I'll leave all of that to you, but it's stable because you have two eigenvalues, both of them have negative real part, right? So indeed, there's one and only one stable fixed point in this setting, and that's the coexistence fixed point. And so if we were to draw sort of flows now on this phase portrait, what you would kind of see something of this type. Is everyone familiar with the kind of what I'm drawing here when I draw these curves with arrows? If you start off at any one of these points, this is marking out a trajectory of where you're going in N1, N2 space over time. The arrows are telling you where you go. So based on where you kind of start off, you'll flow in different ways, but you'll kind of always flow to this coexistence fixed point. There's one and only one fixed point in this entire setup. Okay, is that all clear? I know we took a lot of time to go through this example, but based on sort of how things are going, I can see that this is important. Yeah? Okay, so just in this example, I just want to recap what we did. We started off with a particular situation of this two species competitive Lotka-Volterra model. We wanted both the A's, both the interaction strengths to be less than 1, but of course positive since we're talking about competition. We looked at the fact that there are 4 fixed points. We found that 3 of them are unstable just by either inspection or invasion analysis. And then the 4th one, we looked at the Jacobian and its eigenvalues to figure out that it should really be a stable fixed point. Yeah? So we've talked about stability. Are all these 4 fixed points, by the way, feasible? By feasible, remember we were talking about at the beginning of the class. Something not feasible would be a situation in which at least one of the species abundances are unphysical, negative. That's not the case for any of these, right? So all these 4 fixed points are feasible, but not all of them are stable, right? So this is feasible but unstable, feasible but unstable, feasible but unstable, feasible but stable. Yeah? This is not always going to be the case. We're going to do now another example in which you will see that that distinction really matters. So let's do that other example. So is everyone okay with this coexistence example? Okay, so let's just do an example which is basically very similar, but a little bit of a twist. Yeah. Do A12 and A21 change with time? No, they don't. So does A12, A21 changing with time, would that be like evolution? I would say no, it would be more like a changing environment. Evolution would really be the idea that you get new individuals, for instance, mutants, new diversity that arises, and that sort of plays out. So that really is about changing the number of degrees of freedom of this system. It's not something that usually happens in other physical systems that we're used to. So that's why maybe you're thinking the usual thing about, oh, I just change my parameters with time. But that fundamentally is not evolution. That's actually just changing an environment. Evolution is really changing the size of the system. A12 and A21 are the strength of competition that species 1 and 2 enact on each other. Yeah. All right, so let's do now an example where A12 and A21 are both going to be more than 1. And what should we expect in this example based on this diagram that we've drawn here? Bistability, right? Okay, so this one should go fast. Hopefully we've all built up a lot of intuition by spending a lot of time on that example. So how many fixed points should we expect for this situation? Ready, 3, 2, 1. Okay, everyone's a bit lost and confused. Do you need more time? Okay, let's take 10 seconds. Remember, the equations are the same, just A12 and A21 have changed quantitatively to become larger than 1. Sorry. Okay, ready? 3, 2, 1. Okay, so most of us think there are 4 fixed points. Some of us don't want to participate at all. Okay, and which 4 might they be? Basically the same ones, right? The 0, 0 is still a fixed point. 1, 0 is still a fixed point. The equations didn't really change at all, right? And maybe you guys are worried about this 4th fixed point over here. I mean, algebraically, it will still remain this. That's, yeah. But I guess now we can ask ourselves, do we expect it to be feasible or unfeasible? So we'll vote again. Feasible will be thumbs up. Infeasible will be thumbs down. To remind you, feasibility would be both species abundances should be non-negative. Okay? So in this situation, this 4th fixed point over here, is that feasible or infeasible? Ready? 3, 2, 1. Okay, most of us think it's feasible. And why is that? 1 minus A12 is negative, but 1 minus A12, A21 is also negative. Negative over negative is positive, same thing here. So it's a feasible fixed point, right? So it lies somewhere here in this first quadrant. So 4 feasible fixed points. Okay, so are we basically going to get the same picture as we got here? The null lines are going to still look the same. What's going to happen in terms of this picture to the null lines? So compared between here and here, A12 and A21 became larger than 1 now, right? So would anything change about these? I guess these two null lines stay the same, right? What about these two null lines? What about this? Let's look at this green null line. I guess it will still have this 1 over here, but what will happen to this guy? It'll kind of curve in, right? And same thing here. This guy over here, this will still remain at 1, 0, but this will also curve in, right? Because 1 over A12 is now less than 1. Yeah? So you'll still get an intersection, but basically the orange and the green lines will flip. Right? So right now, orange is above green, but now green will be above orange. Oh my. I really don't know how to make lines. Okay, let's try this again. And this guy over here should give me something like this. Yeah, is everyone okay? To remind you, orange is n1 dot equals 0 and green is n2 dot equals 0. I haven't made the other null lines, they will kind of remain the same. Yeah? Is everyone okay with this? So let's just quickly go and do sort of stability analysis. Is the origin stable or unstable? Ready? 3, 2, 1. It's still unstable, right? You still perturb with a little bit of n1. Nothing changed about the fact that it's still basically a logistic model here. This is 1, this is 1. This is 1 over A, is it? 1, 2. This is 1 over A21. Okay, so this one is unstable automatically. It's also unstable in this direction. What about this fixed point, 1, 0? Stable. Okay. Why do you think it's stable? So I guess you're thinking about now doing the same invasion analysis as before, adding a little bit of species 2. You go back to the same thing that you had over here. Maybe I'll write down the invasion condition again. So you add species 2, look at n2 dot. n2 dot is n2 minus 1 times 1 minus n2 minus 1 A21 n1. n2 is very small because you are doing an invasion. n1 at this fixed point is 1. So you end up with 1 minus A21, and that's negative, right? Because now A21 is larger than 1. And so indeed, if you try to now invade with a little bit of species 2, you actually come back. Same thing here, right? You add a little bit of species 1 and you come back. Of course, that doesn't prove that it's stable. Maybe there's some secret line here along which you'll sort of run away. So how do you check that? Look at the Jacobian, not now around that coexistence fixed point, but around 1, 0. Same thing here, now over 0, 1. And I leave this as sort of homework. You can again kind of check that these are now going to be 2 stable points. And what will happen at each of these stable points? One of the species wins, the other one goes extinct, right? But there's still a possibility of coexistence. So what happens to this guy? Is it stable or unstable? Ready? 3, 2, 1. Unstable, right? Why is that? Yeah, I guess we can now just go back to this picture. The trace is still positive, negative, but the determinant, which had this 1 minus A12, A21, now becomes negative, right? So we end up with a saddle, right? So we also get to know a little bit what this instability is like. And what is a saddle typically? Do people know what a saddle is? There's one direction that's stable, and indeed you can kind of work it out that it will be this direction. So this is actually going to be a stable direction. Along this line joining the origin and this coexistence fixed point, you will actually come and get attracted to this fixed point. But then basically every other direction, you're going to run away. Not doing a very good job at kind of making these flows accurately. But that's kind of what is happening. So really what is happening is that there are two stable fixed points separated by what's called a separatrix. That sort of joins the two different basins of attraction, right? There's one region here, which is called the basin of attraction for species 2 winning, because anywhere you start in this region, you'll get attracted finally at long time to a solution where N2, species 2 is the only one that's surviving. Anywhere you start on the right of this line, you will get attracted to species 1 winning, right? So you can kind of see how big the basin of attraction for species 1 is versus 2 is, and that will depend on the slope of this separatrix line, right? You know the slope of the separatrix line is going to depend on this coexistence fixed point, which depends on the interaction strengths, right? So just by tuning the interaction strengths, you can tune that separatrix and you can tune the relative likelihood that species 1 or species 2 wins, right? In other words, you can tune what that critical fraction is of species 1 versus 2 that you need to add so that you'll get attracted to species 1 versus species 2. Yeah, there's going to be one more situation that we can talk about quickly but not do fully in the next 5 minutes, which is just this last situation of where there's exclusion. Right? So I just want to draw that out because we just see it's a little bit different. So let's do a situation in which A12 is less than 1 and A21 is greater than 1. Oh, sorry. I want to do the other way around. Okay, what should you expect in this situation? Species 1 suppresses species 2 less than species 2 suppresses 1. So what should you expect? Species 2 wins, right? This is exclusion where 2 wins. You can indeed kind of, we're just kind of verifying that this kind of diagram is true, right? And you can also see why 1, 1 was really important. In everything we are doing, the comparison of this A12, the right scale is always this one. Yeah? Okay, so let's just do this out quickly. You know what question is coming. Maybe you're already prepared for it. What's the question? How many fixed points, right? Yeah, you're on the ball. Okay, so how many fixed points in this situation? Ready? 3, 2, 1. All right, everyone thinks it's 4, and which 4? Same story. Now it's getting boring, right? Origin. Oh, by the way, are all 4 of them feasible? Right, so the 0, 0 is still a fixed point. 1, 0 is still a fixed point. 0, 1 is still a fixed point. There's a 4th one, which used to be where? Well, we used to say these two null lines where they intersect, right? Do they always have to intersect? I guess unless they're parallel, they do. But do they always have to intersect in this first quadrant? No, right? You might have a situation in which they actually intersect in a way. Let me see if I can draw this well. Probably going to do this bad job. I don't know if this is accurate, but I just want to show you this, that you might have a situation in which these two null lines are actually intersecting, but somewhere here, right? So mathematically, that would still be a fixed point, right? So maybe your 4 is not technically wrong, but it's not a physical fixed point that you ever expect to see given the dynamics, right? And actually, if you do run these dynamics, you start off with positive abundances, you will never go to a situation where the abundances become negative. Never ever. Outside of if you were doing the simulation wrong. So this is a fourth fixed point, but it's infeasible. Maybe I drew it out wrong in this example. That's what I was saying, that maybe I've drawn it for the other way around. But one of them will be negative. Yeah, so this I probably, like I said, I didn't do very carefully. But I think that's the only thing I wanted to point out, that you'll still have 3 fixed points that are as usual, and a fourth fixed point that is infeasible. So there's no point talking about its stability. So you really only probe the stability of these 3 fixed points, and you will find that 2 of them, the origin will always be unstable, and of these 2, one and only one of them will be stable. You can check that with the Jacobian, and that will be one that corresponds to 2 winning. That would be this one. Again, like I said, I probably did this drawing wrong, so don't let it mislead you. Yeah, and the other example, the whole thing will flip. Yeah. Yeah. So when you're at the origin, right? So what do you think happens at the origin? Both species evolve the same. What do you mean by that? Yeah. Both of them are basically, it's just like saying this is 1 minus n1 minus n2, right? So this is just kind of, you colored the same species 2 different ways, right? Both species are identical. You kind of don't care if there's more of species 1 or more of species 2. It's a neutral situation in which it's just like taking 2 of the same sets of things and just coloring one red and coloring one blue and calling one 1 and calling the other 2, right? That's kind of a degenerate or neutral case. Okay, so we will wrap up here for today. To remind you, we looked today at only low dimensional models, but we did so really thoroughly. We looked at what happens and how you do experiments where 2 species compete. If you know how to do this, you can imagine how you would do experiment where many species compete. Just instead of adding 2, you add many. We saw that usually leads to 3 outcomes, and those are exactly the 3 that you can produce with this 2 species Lotka-Volterra model, where there's just competition. And on the way, hopefully we learned a little bit about how to analyze dynamics of, to analyze nonlinear dynamics of this kind. Really think carefully about feasibility and stability, as well as a fixed point structure. And all of this is going to be important tomorrow onwards when we start talking about the situation where there are infinitely many species. So tomorrow, we'll essentially do the same thing that we do today, except we're not going to be able to make any phase portraits anymore because there are going to be infinitely many species. And we're not going to be able to tell each other exactly whether A12 is greater than 1 and A13 is more than 1 and so on and so forth. We're instead going to take a statistical approach to the interaction strengths. And then, but we're going to ask the same set of questions. How many fixed points? How many are stable? What's the structure of the ecosystem at some typical fixed points? Okay? All right, and we'll see that what we'll need to do that is techniques from the physics of disordered systems. In particular, what we'll do tomorrow is something called the cavity method. Okay? All right, I'll see you all tomorrow.